So what is expected is regarding the ultra structure of a muscle. Basically, you have all studied about the different types of muscle. There are three different types of muscles are there in our body. One is cardiac muscle, skeletal muscle, and the smooth muscle. Out of this, skeletal muscle is the most widely distributed or most abundant type of muscle, representing nearly 40% of our body weight. So wherever our bones are there, bones are always associated with a type of muscle which we call as a skeletal muscle. So they are always associated with the skeletal system. That is why we call it by the name skeletal muscle. And there are certain types of muscle which are present in our stomach as well as in our intestine. And that type of muscle is known by the name smooth muscle. And thirdly, some type of muscles are also associated with our heart or our cardiac system, and they are known by the name cardiac muscle. These are the three basic type of muscle that we can see. So if you take any muscle, especially if you are taking a skeletal muscle, you can see that this skeletal muscle will be this is the entire muscle. This entire muscle will be usually connected with the bone by means of a structure known by the name tendon. Now what you are seeing in the picture is that this is the entire muscle, and there will be an outer covering for that muscle which we call by the name epimysium. Okay, so the epimysium is the outermost covering for a muscle. Now if we look at inside, on the inner structure of the epimysium, we can find a bundle of structures. See, this is one bundle, this is another bundle, this is another bundle, this is another bundle, and these muscle bundle are called fascicle or muscle fascicle. Okay, so here we can find 1, 2, 3, 4, 5, 6 fascicles are there. And if you take one fascicle, again that fascicle will be covered by a layer, and that is known by the name perimysium. See, so this is the outermost covering is known by the name epimysium, and here there is a covering. That covering is known by the name perimysium. Okay, that is the second internal covering. Now again you can see that if you take one muscle bundle, inside that muscle bundle we can again see that number of smaller structures are there, and these smaller structures are known by the name here there may be a better picture here somewhere here. Okay, so this is another better picture. So this is a muscle and it is connected with the bone by means of a tendon. This is the entire muscle, and these are the different types of fascicle. And if you take one fascicle, actually there are still smaller structures, and these smaller structures are known by the name the muscle fiber or muscle cell. That is, inside each muscle fasciculus we can find number of smaller elements which we call by the name muscle fiber, otherwise known by the name muscle cell. So basically the term muscle fiber and a muscle cell are the same thing. So what you are seeing here is a muscle cell, or we can also use the term muscle fiber. Okay, so and each muscle fiber will be having a nucleus, there will be a cytoplasm, etc. And different cellular structures will be present inside the muscle cell. Now one more important feature of a muscle cell is that inside the muscle fiber or a muscle cell there will be smaller structures. They are almost thread-like or fiber-like or hair-like structures are there, and they are known by the name myofiber. So a muscle cell or a muscle fiber will be in turn formed of several myofibers. So this is how the structure of a muscle fiber, skeletal muscle is arranged. On the outermost we are finding the entire muscle covered with the epimysium. Then we have muscle bundles which we call fasciculus. Each fasciculus will be covered by a perimysium. Then we are seeing that inside each muscle bundle or muscle fasciculus, we can find several muscle cell or muscle fibers, and each muscle cell will be in turn formed of numerous smaller filaments known by the name myofiber. So this is the entire arrangement of a muscle fiber. Now what we have to see again, maybe you have studied this in your +2 classes. Now if you take one muscle fiber, we can find that muscle fiber usually having a dark region as well as a light region. And these dark regions are known by the name A band, and the light regions are known by the name I band, otherwise known by the name light band. And the dark regions are known as an anisotropic band, because they will not be reflecting light much. Whereas a light band is known by the name isotropic band or I band, because they will be reflecting more light. So that is how these dark and light regions are present on the surface of a muscle, and this is known as a stripes or striation. That is why skeletal muscle is sometimes known as a striated muscle, also known by the striped muscle. So because of this dark band or white, just like a zebra crossing, you can see the dark regions are there as well as light regions are there. Then you can, what you are seeing here is that if you, this is how the dark region and the light region. Now you are taking, this is the entire muscle cell. What is other name for entire muscle cell? Also known as muscle fiber. Now if you take one muscle cell or a muscle fiber, that will be formed of numerous smaller structures known as myofiber. Okay, now we are taking one myofibril and we are enlarging it. Here you can see that. Now if you enlarge that myofibril, we will be getting a picture like this. Now there are two regions. One is known as Z disc here. That is, each myofibril is actually formed of two kinds of protein. One type of protein is a thicker protein known by the name myosin. So this red colored structure is actually a thicker protein, and it is known as a myosin filament. And there is a blue colored line here which is a thinner protein which we call as the actin filament. So what you are finding here, actually this myofibril is actually formed of two types of protein, myosin protein as well as actin protein. Myosin protein is a thicker protein and actin protein is a thinner protein. That is the difference between myosin and actin. Now we will see how this entire thing is arranged. Here also you can see that this is the myosin and this is the actin. Now here look at that. Between the this is actually this blue line are indicating the actin protein. Now at the center of the blue line we can find there is a darker portion. That darker portion is known by the name Z disc. And similarly here also at the center of the actin protein, we can find a darker region which we call as Z disc. And we are seeing that there are two regions between two Z discs. The region between this Z disc and the region between this Z disc is known by the name sarcomere. The significance of the sarcomere is that there is a fundamental unit of muscle contraction or the basic unit of muscle contraction. Now one sarcomere is again enlarged here. Now what we are seeing inside the sarcomere, we are finding an actin region, and at the center of the actin region we are finding the Z line or Z disc. Now at the middle we are finding the myosin protein, that is a thicker protein. Now if you look at again that at the middle of this sarcomere there is a lighter region, and this lighter region which we are seeing at the middle of the sarcomere is known by the H zone. So H zone is another important structure. So this is the Z line, and the region between two Z line is known by the name sarcomere. And if you are taking the center of the A band, we can see that there is a lighter region. This lighter region is known by the name M line. Why it is lighter in color? Because here we can find myosin protein and actin protein. But at the middle there is only myosin protein. And because of that, the center of the myosin, the center of this A band will be slightly paler or lighter in color, and that region is known by the name H zone, and at the middle of the H zone there is a line known by the name M line. So this is how the H zone and M line are coming. Now we have to also keep in mind what is an Z line and what is the significance of sarcomere, and why the muscle is showing this light band and dark band. And if you look at the closely at the muscle, this, if you take a band is actually this region where myosin is there, as well as some portion of actin filaments are there. And I band will be actually showing this region where only actin filament is there. So the region where actin filament are only present will be shown as the I band. The region where both actin as well as myosin are also present will be shown as an A band or a darker region. So that is how the muscle is exhibiting A band as well as I band, or darker region as well as lighter region. So this is an idea regarding the internal structure of a muscle, and that is and you can see that how now what we have to see is how a muscle is undergoing its contraction. That is another portion that is expected, that is what is the mechanism of muscle contraction. And there is a basic theory known by the name sliding filament theory. Now in case of sliding filament theory, here you can see that now you may be able to understand what is a sarcomere. Now we are going to take a sarcomere in a close-up. Here you can see a sarcomere. What is this? This is the Z line. This is another Z line. And the region between two Z line is known by the name sarcomere. This is the myosin protein and this is the actin protein. You can see that in the middle of the myosin protein there is a paler region. Why it is paler? Here there is no actin filament, and that is known as H zone, and this middle line is known by the name M line. So this entire portion is known by the name H zone. Here you can see that the entire portion is known as H zone. The line that is present in the middle is known by the name M line. So now we will see how the muscle is undergoing a contraction. Actually, the muscle is undergoing a contraction. Here this is, you can see the Z line. Now you can again see the internal structure of a muscle, how the different structures are arranged, and these are all myofibrils. You can also see that. Now there may be video. I don't think this video will play. If it is playing, I will. Yeah, you can see that this is a video. See how the muscle anatomy muscle will be contracting and relaxing. You can see that. This is a slow video. See, okay. Watch this video. You can see how a muscle will be contracting and what are the basic parts of a muscle. This is the individual myofibril. Now you can also see that this is one myofibril, and each there will be covered with the endoplasmic reticulum, that is known as a sarcoplasmic reticulum. We are going to get a close-up of that, and this is the myosin filament, which we call as the thick filament, and this is the actin filament, which we call as the thin filament. Now we are taking a cross section, and we will see how the entire portion is arranged. can see that this is the eye band. The light regions are known as eye band, and you can also see what happens during a muscle contraction is actually this length. There is no physical contraction in the length of the A band or I band. It is basically moving or sliding of A band into I band region, or sliding of I band into the region of A band. So this is how the muscle is actually undergoing a contraction. See, you can see that when muscle can only contract, it cannot extend. So extension is simply a passive process of relaxation. Now you can also see that the muscle will be having a signal system. When a electrical message or message is reaching there, calcium ions will be released, and that will be causing the reduction in the length or decrease in the length of the muscle fiber. So when we get a nervous message, one calcium ions are released, and this releasing of calcium ion is what is causing the muscle contraction. So this is an electron microscopic, this is a microscopic view, probably electron microscopic figure. So here you can find that this is the myosin and this is the actin filament. And what is happening is that this actin filament will be moving into the middle of the myosin filament. So that is what is happening in a muscle contraction. You can also watch that. And this is the M line, and this is the. So look at the picture. You can see that. So you can see that this portion will be coming together into the middle. So here now look at that, how the length of the sarcomere is. So this is actually muscle contraction. And during a muscle contraction, this there will be moving to, just like our fingers are moving into each other. So the overall length of the muscle will be decreasing, and this process will be repeated in several sarcomere. So this is how the muscle is actually undergoing the contraction. Here also you can see that there are certain protein structures which are helping in this contraction, and these are known as the myosin head. They are actually protein and they are actually pulling it. So this is known as the myosin head, or this is actually a protein. They will be attaching it and pulling it using some using energy released by the ATP. So this is the, for this attachment, energy is needed. Okay, for this attachment of myosin head with this actin, some amount of ATP or some energy is required. Only by utilizing the energy, this attachment, in order to attach and pull it like this, they need some energy, and ATP is supplying that energy. You can see that this head will be pulling the structure in this direction. It will be pulling like this. So it is by this pulling mechanism of the myosin head, actin is coming closer. So now we think it is a very simple thing, but actually to discover this all process, scientists have taken several years. Lot of technology have been utilized here. See, this is how we can see that will be attaching it and pulling it. So it is how the muscle will be contracting. For each process, energy is utilized from the ATP. So this process is what we call myosin cross-cheek, and ATP will be splitting, forming energy, and this is known as the myosin head. So this is how the muscle will be undergoing its contraction. When a muscle is undergoing contraction, the length of the muscle will be decreasing. And this process is known by the name sliding filament theory. So this is what is explained in the sliding filament theory. You can see these are the myosin head. You can also see that there will be here also you can see that this is a relaxed muscle, and what happens during a muscle, this is the relaxed state. When a muscle is contracting, Here you can see that these actin filaments will be coming closer into the middle region. In the next region, what is happening? They are again coming closer. So you can see the picture. This is the initial stage, and during the second stage, the muscle will be partially contracted, and during the third stage, you can also see that the muscle has undergone full contraction. So this is how the muscle is actually contracting, and the theory that explains this known by the sliding filament theory. Why it is known as a sliding filament theory? You can notice that there is no decrease in the length of the myosin protein, or no decrease in the length of the actin protein. The only thing that is happening is that actin will be moving into the region of the head, and just like our fingers are moving into one another, the actin will be moving into the region of the myosin, and the overall length, earlier the length of the muscle was this much. Now because of this process, the overall length of the sarcomere has decreased, or overall length of the muscle will be decreasing. And this process is known by the name sliding filament theory. And what you are seeing is how the crossbred, this structure is known as the head of the muscle. Using the energy released by an ATP molecule, the ATP will be splitting. It will be pulling this actin filament in one direction and causing this decreasing the length of the sarcomere. So this is how the whole process will be going on, and this is known by the name the sliding filament theory. And sliding filament theory is actually helped by the release of ATP molecule. So here you can again see that what we have just now seen is the head of the myosin, and they are undergoing, and this theory was actually put forward by two scientists known by the name Huxley and Hansen. And I-band means the lighter region, and H is on. So contracted muscle becomes shorter and thicker, and but the volume will be decreasing. Now here you can also see what happens to the energy. You can see that there is a splitting of ATP, and in presence of calcium ions and magnesium ions, an enzyme known by the name myosin ATPs will be there, and that myosin ATPs will be undergoing, will be responsible for the splitting of ATP, forming ADP, inorganic phosphate, and energy. And this energy will be utilized for this muscle contraction. Here you can see that in order to attach this head with the actin filament, energy is required. This energy is actually supplied by splitting of ATP molecule. Here also you can see that what is happening, and with the ADP will be combining with another creatine phosphate, forming recreating the ADP again, and for that there is an enzyme known as creatine kinase will be utilized, and ATP will be again recreated along with the creatine molecule. So this is how the process of muscle contraction is actually what is the chemistry behind the muscle contraction is what is explained here. ATP will be undergoing splitting, forming energy and ADP, and ADP will be again combining with the creatine phosphate or phosphocreatine, recreating one new molecule of ATP along with the creatine. You can also so these are some of the other changes that are happening. Now according to your syllabus, you have to also study another area known by the name muscle fatigue, tetanus, and rigor mortis. Now I think in this section it is explained what is a muscle fatigue, also explained here. You can see that in case of a muscle fatigue, basically what you are seeing is that when a muscle is continuously stimulated, and the muscle will not be having enough amount of chemicals. Basically this muscle is contraction is triggered by a type of neurotransmitter known as acetylcholine. When acetylcholine is decreased, so muscle will not be having enough amount of acetylcholine, and muscle will not be responding to any stimulus. That situation is known by the name fatigue or muscle fatigue. So muscle fatigue is also an important process that is happening during muscle contraction, and this muscle fatigue is actually caused by the decrease or shortage of acetylcholine. Acetylcholine is the. Then another peculiarity is that you might have noticed that when a person is dead, the muscle becomes stiffer. If you touch any dead animal, you can feel that there is a stiffness or hardness to the muscles and joints of a dead body. And that situation is known by the name rigor mortis. Rigor mortis is actually caused by the stoppage of aerobic respiration. So in the absence of aerobic respiration, the muscle may not be undergoing relaxation after a contraction. So muscle once contracted, it will remain in that contracted position. In order to relax the muscle, they need oxygen. So when no more oxygen is getting, this splitting of ATP will not be taking place and muscle will remain in a contracted state. That situation is known by the name rigor mortis. So rigor mortis is actually starting after 10 minutes after the death, and after 24 hours the rigor mortis can and undergoing a there is a process called decomposition will be starting. Now after 24 to 48 hours after the death of the person, rigor mortis will be slowly decreasing, and that is because of the decomposition or decomposition, and that process is known by the resolution of the rigor mortis. So this is what is explained here. Now this is so remember that this ultra structure of the muscle, that is an important topic. You have to keep that in your mind. Then you have to also study what you mean by sliding filament theory, and that is also an important topic for your examination. So we will be stopping here. We have already 12:15, and I will give some questions also.